Thank you for watching this training video about product and environmental sampling methods. In this training session, we focus on the sampling methods and proper handling of samples. If you need assistance in developing a sampling plan, please contact AEMTEK for consultation. At AEMTEK, we strive to provide accurate, fast, and reliable analytical services to our clients. Results of laboratory testing are limited to the exact samples provided by the client. So proper sampling is a prerequisite for accurate laboratory testing and meaningful results. Therefore, by improving sampling procedures, we can improve data quality. First, let's discuss the basic concepts of what a sample is and how they should be taken. By definition, a sample is a small portion of the lot or the environment in which the distribution of both number and types of microorganisms should be representative of the whole lot or the environment. This is only true when microorganisms are homogeneously distributed, such as in a well-agitated fluid product. But in general, the distribution of microorganisms is presumed to be random. In nature, the microorganism distribution is often heterogeneous. For environmental testing, the seek and destroy strategy indicates to sample the sites that are likely to be contaminated. There are 3 steps associated with sampling. The first step is to have a sampling plan that is developed based on risk assessment, processes, and resources. A proper sampling plan should, at minimum, specify what to sample, where the sample should be taken, when and how often to perform sampling, and how many samples should be taken. Please reach out to AEMTEK Laboratories if you would like us to help with developing or reviewing your sampling plan. The second step is the physical collection of samples. You will need proper containers and tools to take the samples, making sure you take enough samples, and following correct methods and procedures to collect samples that are suitable for analysis. The third step involves proper documentation of the sampling event, so everything that occurred is clearly understood. This includes labeling each sample with unique numbers, proper storage of samples before dispatch so they don't spoil while in transit, actual transportation of the samples to the lab, and proper reception of the samples by the lab. All steps must be controlled and properly documented to ensure the quality of the samples and the test results, and to allow reliable traceability. Various tools or utensils can be used to take samples. Spoons, cups, knives, scoops, or pipettes can be used to collect food samples, or sponges can be used to collect environmental samples. Various containers can be used to contain samples once they are collected. The common ones include plastic bags, bottles, jars, boxes, etc. No matter what shape or size the tools, the ones used for sampling for microbiological testing should be sterile, or at least cleaned with a disinfectant and then rinsed thoroughly. Because if the tools are not sterile, the sample can become contaminated and the results will not be accurate. There are a few basic requirements when collecting samples. First, you need a sampling plan that fits the purpose. Sampling plans should be scientifically backed and based on risk assessment of the specific product, process, and environment of your production. Samples should be representative of the lot, and environmental sampling sites should be determined with the intent to seek and destroy pathogens. Second, those performing the sampling must be trained and instructed properly on collecting samples, specifically on aseptic techniques that are crucial for microbiological sampling. Third, the sampling method and environment must be safe for the sample collector. And last, the sampling procedures must not allow contamination of the food, environment, or personnel. Aseptic technique is crucial to microbiological sample collection. Aseptic technique involves target-specific practices and procedures under suitably controlled conditions to reduce the contamination from microbes. In short, aseptic technique is used to ensure that the sampler is not introducing any foreign microbes to the environment that is being sampled. Using aseptic technique when collecting samples is an essential element of good microbiological laboratory practices, or GMLPs. Aseptic techniques for collecting food samples include washing hands before you go to collect samples, wearing the appropriate personal protective equipment, or PPE, and changing gloves often and when dirty or torn. Using sterile containers or bags and not exposing them to the environment before sampling, not touching samples with bare hands or dirty gloves, not touching the inside of the sample collection container or bags, and washing your hands immediately after removing gloves. Aseptic technique for collecting environmental samples includes all the techniques from collecting food samples and sanitizing the food contact surface after sampling, securing the sample container or bag, sanitizing the exterior, and ensuring the samples are labeled properly, and ensuring engineering controls such as eye wash stations, safety showers, and sinks are properly maintained and inspected. We always want to think of safety as the first priority. Let's discuss the different types of sampling procedures in some detail. Sampling of products should follow the company's sampling plan. Routine samples can be taken at defined intervals during the process. For example, at the beginning, middle, and end of a lot for every 2 hours of production run. Samples can also be taken at an event, such as after startup, after stops, or after changing something. Some samples can be taken from pallets according to the sampling plan. And the sample collector should always take caution to clean and disinfect the external surface of the package and open the packaging materials properly to avoid contamination. While collecting samples, it is important to keep a few details in mind. We want to be sure we're collecting enough sample for analysis. A good rule of thumb is to collect twice the amount of volume that is recommended for testing. This will ensure there is enough sample in case additional analyses are necessary. If a package shows signs of swelling or spoilage, send the intact package for laboratory analysis. This will ensure the integrity of the environment within the package so we can determine what is causing the spoilage. Always use sterile sampling tools while collecting samples. And ideally, food samples should be mixed with a sterile tool to ensure a representative sample was collected. The tool used to collect the samples must be sterile so that the sample is not contaminated. The chain of custody form, or COC form, is the order form for laboratory testing. Sample information will be transcribed to a certificate of analysis, or COA, as a permanent record. So it is very important that the information on the chain of custody form is accurate. For all chain of custody forms, contact information must be completed. Project information details can also be provided at the company's discretion. For product samples, a lot number, production date, lab ID, or other identifier must be included. A sample description or other notes must be provided as well. It is important that the combination of the client sample code and sample description are unique for each sample provided, so that there are no repeated code numbers and the results will make sense. For environmental samples, information should include the type of sample and the description of samples, including location where it was taken, sample volume, and other details. We'll cover how to properly fill out specific chain of custody forms later in the video. For sampling potable or processing water, perform sufficient flushing before sampling. Use a sterile ladle for water or solutions in open systems. If necessary, use neutralizing buffer to neutralize disinfectant residues. Please visit the AEMTEK YouTube channel for instructions on how to take potable water samples. For water samples, it is necessary to record the date and time when the sample was taken on the chain of custody form. In addition, water samples should be kept cold after sampling and before testing. Always remember to have a unique sampling description for each sample. For water samples, it is important to get them to the lab as soon as possible so that we can begin processing them within 24 hours of collection. Please visit the AEMTEK YouTube channel for instructions on how to take exposure plate samples. Please visit the AEMTEK YouTube channel for instructions on how to take active air samples. Please visit the AEMTEK YouTube channel for instructions on how to take compressed air samples. When filling out the chain of custody form for air samples, please indicate if the samples are compressed air or exposure air samples. Also indicate the time for exposure air and pressure for compressed air. Always remember to have a unique sample description for each sample. Please visit the AEMTEK YouTube channel for instructions on how to take surface samples. There are a few important details to keep in mind while taking environmental surface samples. Sponges are used to collect samples to test for the presence or absence of pathogens on a surface. Be careful while sampling food contact surfaces. Don't leave any residues on the surface and sanitize after collection of the sample. Also keep in mind that you can sample a large area with one sponge, but never sample across zones. Swabs are used for enumeration of indicator microorganisms. For this reason, swab within a defined area, for example 4 square inches. It is important that this be a standard across all sample collectors within your plant. Sample collection sponges and swabs should contain a neutralizing buffer to neutralize any disinfectant residues left from sanitation. Do not leave the handle in the bag. The handles should be removed from the sampling sponge aseptically. After collecting an environmental surface sample, hold the outside of the sponge bag and place the sponge end in the bag, being careful to not put your fingers in the sampling bag. Once the sponge is fully in the bag, using the hand that is on the outside of the bag to hold the sponge while you disconnect the handle. Then close the sample bag. The key is to not allow your gloved hands or any part of the handle that have touched your gloved hands into the bag. Lastly, put your collected sample into refrigeration as soon as possible so that no microorganisms can grow and alter your results. When filling out the chain of custody or COC form for environmental surface samples, please indicate if the sample is from a food contact surface, if it was collected post-sanitation, if it was collected using a swab, single sponge, or dual sponge, and the date that the samples were collected. Always remember to have a unique sample description for each sample. While labeling samples, write numbering and/or descriptive terms directly onto the sample container or bag using a Sharpie or secured label. Use sequential and unique numbering for each sample. Numbering and descriptions on the physical sample must match the chain of custody form. Verify your chain of custody form to be sure that these match. An internal sampling report is important and should include the date and time of sampling, who took the samples, the sample identification and description, and any special observations. Schedule transport to the laboratory as promptly as possible. Storage conditions should be stable and must prevent changes to the samples, especially microbial growth or death. Keep samples refrigerated or frozen as necessary. Before sending samples out, check that your physical samples match the chain of custody form. Be sure that the samples are packaged to prevent breakage or spillage of the samples. Upon receipt to the lab, AEMTEK laboratory technicians inspect the integrity of the package containing samples. Temperature checks are performed on samples that require refrigeration or freezing. Our technicians then perform a cross-check against the sample report or chain of custody form and physical samples. Our account manager contacts clients if there are any discrepancies. Please ask your account manager at AEMTEK for login instructions to access your results data online in the LIMS program. We hope that this sampling training was helpful and that you feel prepared to take environmental and food samples at your plant. Please don't hesitate to reach out to your account manager with any questions or concerns. We are here to help you.